everyone in this room knows Dr. Gotch is a living legend. Dr. Gotch graduated from Berkeley and then did his medical school at UCSF. And he did his subsequent residency and internship at all the three San Francisco General Hospital, UCSF, the VA, and the Moffitt. There was no renal fellowship back then. And today he's going to come talk with us about the early days of hemodialysis at San Francisco General Hospital and Davis. I've been trying to get him to come speak with us for several years, and I'm really happy that he's able to come. And please give him a great round of applause.
Thank you. I hope he said nice things because I cannot hear him. Well, what I'd like to talk about is essentially the history of dialysis in San Francisco, and particularly how it started and the early years. It all began when John Carboni, who had been my chief resident as an intern, asked me if I'd set up a artificial kidney at San Francisco Hospital. My mentor, and at that time hero, Izzy Edelman, advised me not to get involved with dialysis. In his view, the kidney had now had a trial and it had no future in the management of renal disease. And to the best of my knowledge, Izzy never changed his mind and never really believed there was a role for dialysis in academic medicine. It's wonderful to see that you now have a chair in bioengineering here. So I'd like to talk about dialysis in the ICU at San Francisco General in the first 4 years to begin with. This is where we started. This was a terrible device. The twin coil kidney. We set it up for treatment of acute renal failure and poisonings. And we had nothing other than a brachial or radial artery cut down for access. So until Quentin developed the cannulas, we could only treat a patient 2 or 3 times because you'd run out of sites to do cut downs on. Then a very short time later, the cannula system became available. I learned how to bend the Teflon with a blowtorch in my garage. So we assembled those and brought them in to implant for patients. So now we had access for several dialyses, theoretically an infinite number of dialyses. Now the twin coil dialyzer was, as I said, just terrible. You couldn't measure blood flow. You couldn't control ultrafiltration. You couldn't control clearance. You never knew whether you dialyzed the patient or not until afterwards you'd get the chemistries and the weight. And sometimes you'd take off a lot of weight, other times nothing. And sometimes the B-win would fall 10 or 15 milligrams per deciliter, and other times there'd be a big drop. It was a totally unpredictable, gadgeteering designed device. Now, a very critical point in this history is the relationship between Donna Mapes, who I recruited as our head nurse, and a California Assistant Attorney General, and the Assembly Bill, which enabled the 2 dialysis centers in 1966. We had the Attorney General referred to us with acute renal failure, the cause I can't remember now, and he'd had a very severe toxic psychosis. And I was doing something, I looked around, and he'd gotten loose from the restraints. He had his legs around Donna's neck. She was turning blue, and we managed to separate them. And he did recover, and he gave very valuable testimony on the floor in Sacramento that I'm sure played a significant part in the passage of this Assembly Bill. Now, by '66, Scribner and people in Seattle had solved the logistics to enable chronic dialysis therapy. And he also played an important role politically in getting the funding of chronic dialysis therapy. And 2 dialysis units were established in Northern California and Southern California. Donna and I put together a proposal for the one here, and since we were the only kidney center around, we got it. And this was the workhorse dialyzer that was used at that time, the Keel. Again, it was designed and built by a creative physician, and it was not very efficient. Clearance of 80 or 90, weighed 60 pounds. You had to build it by hand, and it was not very reproducible. Now, the other big thing that happened about that time, within a year of getting the enabling of the 2 units in California, was the development of the NIH Artificial Kidney Chronic Uremia Program, and the conjunction of Dow Chemical Company being just across the bay in Walnut Creek. This program resulted in a number of grants for development of all aspects of dialysis therapy, and Dow was granted an award to develop the hollow fiber kidney, and we were here, and so we got a contract to work with them on this. This, I really think, was the golden years of hemodialysis research. This program made available funds only for dialysis in the order of $5 or $6 million a year, which was a lot of money that many years ago. And this is someone who played a major role in my life, Ben Lips, chemical engineer, fresh out of MIT, working at Dow, and he was been my personal tutor in chemical engineering, developing the hollow fiber kidney, and many, many problems since then. And I'm going to tell you why I think this is an important relationship. Here's a overview of what the kidney program, artificial kidney program, did. They developed funding for membranes, hardware, cannulas, uremic toxicity, adequate of dialysis. We had our own bank account in Bethesda for funding in this area. So a lot of exciting things were done at that time. Now, the point I want to make with regard to Ben and generally is that hemodialysis research has been a long and highly productive tradition of collaborative research in chemical engineering and nephrology, beginning with Hans Burke and Pim Kolff, and I'll show that relationship in just a moment. Ed Leonard and Bill Blumley, Les Babb and Belding Scribner, Lee Henderson, and he had some really great people, Ellen Michaels, Clark Colton, Michael Lyset. And I was a very slow learner and lived a long time, so I had a really lot of people that taught me about this. Now, I'd like to give you a very brief history of the evolution of thinking about dialyzer solute transport. It began in the mid-1850s with Graham, a Scotch physician who did a lot of very careful, meticulous work in the separation of gases and chemicals by diffusion. He coined the term dialysis, and so and he also separated isotopes of uranium. So in many ways, he might be considered the father of dialysis and the atomic bomb. Science doesn't distinguish between good and bad necessarily. Now, 5 years after Graham did all this work, Frick came along and put together a mechanism to explain what Graham was seeing. And the diffusion constant and first law of diffusion which was later transformed into permeability constant for dialyzer. And essentially, he used a lot of Graham's work to verify these concepts. Then 75 years later, after Graham, in fact, Haas in 1926 and in 1928 developed a kidney which was again not guided by quantitative science but by a gifted physician. And he did do a mildly successful dialysis in 1928. It's interesting to think what a terrible time that was with Hitler and the rise of, in Germany, what was happening, and here he was beginning dialysis therapy. Then came the man that almost everyone is aware of, 100 years after Graham and about 20 years after us. Now, I'd like to use Kolff and Burke to illustrate the importance of engineering and clinical collaboration in the developments in this field. This is the idea that Kolff originally had for the kidney. Tubes of sausage casing laid out in parallel in a bath that was flowing countercurrent. Now, this design would almost certainly have failed. It was simply not a way to get good mixing and to get good conditions to transfer solute between the buds of the dialysate compartment. This is in fact the kidney that was evolved, and Burke was the guy who was behind this. They made this rotating drum. They had the tubing wound around it, and the blood was propelled by the rotation of the tube up, and then it would fall down on the other side. And here was the bath that was seeing the rotating drum. The important thing was that this led to mixing in the bloodstream, and they did not fill these tubes, they were half empty. So the liquid film was small and narrow, and there was good mixing. Here you see it schematically illustrated with the blood going down, going up, and the tube going down as it rotates. And then on the upstroke, there would be back and forth flow. So there was good mixing in the bloodstream and also good mixing in the dialysate stream. And this was Burke's design, not Kolff's design. And it represents, I think, the first real fruits of bioengineering and clinical nephrology combined and working together. Then, about 5 years after that, the first literature on science of transport and dialyzers was published by Wolff. And essentially, he developed a very, very useful but very simple term, dialysate, which is now dividing the flux, the amount of solute transferred, by the gradient, now defined as blood inlet minus dialysate inlet concentration. This then can become a constant that can be incorporated mathematically into the mechanism of transport. And this was a very exciting plot of Wolff's, published in 1950, in which he showed as a function of blood flow in the old coil dialyzer, and he calculated the dialysate and showed it displayed as a function of solute, going from little molecules to big molecules. And he found this family of logarithmic functions, the first time that this kind of analysis had been done. The next step was Renken, who was really a great physiologist, ended up in Davis, actually, Davis campus. He published this paper 5 years later, and for the first time, he integrated the Fick diffusion equation for the rotating drum dialyzer. and with this very simple first-order transport equation, and then was able to back out what he called a permeability times area term, or which we'll show in a moment as the overall transfer coefficient times the area. This could be generalized now into a way of designing dialyzers based on quantitative understanding of the mechanisms of solute transport. How publication has changed in 50 years. This was his paper at ASIO, and this was his diagram showing that he was integrating this drop in this concentration profile per unit of length in the dialyzer. But it was a very important paper, the first solution of the Vick equation to dialysis. Then he published this remarkable thing showing the characteristic relationship of clearance, or dialysance, I should say, to flow rate in the coil dialyzer, and showed that as solute weight decreased, this is urea going up to higher weight. Well, this is actually his term, the KOA, the permeability coefficient. You'll see this logarithmic function that is equal almost to blood flow at low flow rates, and that asymptotically levels out to a value limited by the permeability. And this must have been a very exciting slide for him, because he took Wolff's data that had been published 5 years later as a family of curves defined only by solute size, and now could convert them into well-defined mathematical functions, the dialysance as a function of blood flow rate and the permeability times surface area product. I think this must have been a lot of fun making that slide. And I think it's similar to the excitement I recall at San Francisco Hospital when Izzy Edelman, I was a fellow at that time, and got together the first data showing the universal basis of the serum sodium concentration. It was the concentration of sodium plus potassium in total body water. That was a very exciting relationship to see for the first time. Now, the final really important person that I want to mention in terms of development of understanding was Alan Michaels. This paper was published in 1966, the last paper in the program, no discussion of it, but it was a critically important paper because he extended these analyses and was able to develop the relationships to predict clearance and function in all kinds of geometries, countercurrent, co-current, cross-flow, and you now had a blueprint for engineers and physicians to think about and how you should design a dialyzer to be optimally efficient. And this is simply a variation of the Michaels equation that my friend Ben Lipps used in 1967 to calculate the area he'd need in a dialyzer constructed of very tiny hollow fibers. See, he's gone far beyond what Burke did, where he had the rotating drum. He's now distributed the blood in about 10,200 micron fibers. So the diffusion distance is very small and is widely distributed. This is comparing a silicone cast of the cross-section of the blood flow panel in the Keel dialyzer compared to these tiny channels in the hollow fiber kidney. So you could predict this should make a huge change in the efficiency of the device, and you could actually calculate how big a change it would be. And that began a very exciting period of time in my life in terms of research. We had Walnut Creek, Ben Lipps, John Sargent as the engineers, San Francisco, I was working with Marcia Keene, who did her doctoral PhD thesis in nursing while collecting a lot of this data. They would make changes in design. We would give clinical feedback back and forth. And with this kind of a relationship, we progressed very rapidly. This was the first hollow fiber dialysis in 1968. Mary Ann, who was a high school student, went on to college and worked with us in the lab as a research technician later. But this is a picture of the kidney, and here you see it in color. We very quickly went from this huge, cumbersome keel to this tiny hollow fiber kidney, which had clearances of 300 to 400% increase, 90% decrease in blood volume, and 95% decrease in physical size. So it was an enormously rapid advance in technology, made possible by the advent of hollow fibers and the advent of scientific knowledge of transport. But Mary Ann was the first and only patient treated for the first 6 months because we didn't have enough kidneys. They were not able to make them leak-free. So we didn't know that we had a huge problem waiting for us. When at the end of 6 months, they were really making a lot of kidneys. We started 6 more kidneys, 6 more patients, and they all clotted. They all clotted the device. And we really did not know that was around the corner. So we set out on a 5-year journey of trying to solve those relationships based on the understanding of patient potential to clot, surface properties, and geometry. And we studied all of those things over the next 5 years. We found a way to quantify the amount of clotting using the Michaels equations and the loss of volume in the dialyzer. We looked at the patient clotting parameters. We looked at the sites of clotting and had changes in the geometry that we evaluated. Looked at header flow rates, velocity profiles, tube sheet geometry, but all of that really, except the patient, made a huge difference. The rest of it really didn't amount to much. The whole problem was in the blood contacting surfaces. And here you see one example of clotting that was very puzzling when we saw it. Sometimes patients would run perfectly, and after 2 hours, suddenly occlude the dialyzer, pressures would go sky high, and you'd have to stop. And this is an example. We finally caught this culprit in the act. They would form a membranous clot on the inlet header, and that could dissect down and plaster on the tube sheet and completely occlude it. Here you see there's virtually no clotting on the ends of the tubes going into the dialyzer, and this whole thing was here. This was unusual. It was very satisfying when we saw that relationship. Now, this shows how these dialyzers were fabricated 50, 60 years ago. The cleanup after they were made, they were sterilized and rinsed with reverse osmosis water, but they were all exposed to varying amounts of endotoxin and impurities during the course of manufacture. I think one of the most impressive things that Ben did in this development was he attended a lecture by Herb Perkins, who was director of the blood bank in San Francisco, and it was a lecture on intravascular coagulation. Now that's totally different clinically from what you think intravascular coagulation with these episodes of isolated clotting. But he postulated that could we be getting endotoxin adsorbed onto the surface during the fabrication procedures, and that was the cause of the clotting. So we set upon a sequential program of studying that. We chose patients with history of excessive clotting. They were heparinized, and we perfused the bloodlines alone at first to stabilize that. And we had noticed over these 4 years that the first kidney, if there was going to be clotting, always clotted more than the second. And it exhausted some of the patient's thrombogenesis potential. So the experimental dialyzer we wanted to test would be run first, and the other the control kidney second. We monitored continuously with pressure transducers, and at the end of 45 minutes, we removed the first kidney and put on the second kidney and measured the fiber bundle volume. Now this is the plot that's one of the 2 plots that's most dear to my heart. It shows the results of those studies. This was the endotoxin normally produced, this was the sterile water, pure produced kidney, ran first when the patient was most likely to clot. No loss of fiber bundle, no clotting at all. Then we converted the second kidney, and you can see they were all over the map, just what we'd seen clinically. So we knew that for all practical purposes, we knew where this vexing problem was, and we could now take steps to eliminate it. And took about 5 years to do all that. But at the end of those 5 years, we did have, and I'll qualify this in a moment, a non-thrombogenic hollow fiber kidney that was greatly improved. So we went from this in 63 to this in 67 and to this in 72. I look back on that and feel very fortunate to have been able to participate in those radical changes and improvement in dialyzer technology. Now, I'd like to say just a word about dialyzer design issues now. They're very different. And when I say this was non-thrombogenic, I mean, of course, you had to heparinize the patient. You had to really ensure that their thrombus mechanisms were suppressed. We can't do that if we're going to have a wearable artificial kidney. You have to have a device that really is non-thrombogenic and that can be running continuously, probably with sorbents and something that may be able to be implanted. Hemofiltration has also emerged as probably a useful therapy, and we're working and need to look at ways to develop increasing permeable membranes. And nanotechnology is around. But these are major problems that remain to take the next really big jump in therapy. Now, the second topic I'd like to talk about is kinetic modeling of the dose of dialysis. After dialyzer design was optimized, the question became, how should these dialyzers be used? What is uremia? What do we dialyze? What's an adequate dose of dialysis? And again, the Artificial Kidney Chronic Uremia Program set up a conference in Monterey in 1974 in which experts in all of the organ systems affected were assembled and gave presentations and were asked to devise strategies or ways to monitor the effects of different dialysis regimens on this multiplexity of organ system abnormalities. And the fundamental principle was we think that dialysis as uremia is due to the accumulation of body weight, but what solutes? Now, I'd like to very briefly look back couple of hundred years. The search for toxins really began with Prevost and Tomas reporting symptoms of chronic nephritis and elevated blood uremia in nephrectomized dogs. Bostic found then 5 years, about the same time, 5 years later, the same kind of things in patients with kidney disease. But then 10 years later, Bright reported this astounding paper that showed the same levels of BUN and the patients were clinically well. So this was the beginning of the 200-year war of urea, and totally contradictory findings. You can plot that situation here. What I've got on the ordinate is BUN. On the x-axis is an arbitrary well-being score. Dead, perfect health. And here you had classical uremia, according to Bostic, very sick with BUNs of 80. And out here, we had patients clinically well with the same BUN, which were the patients that Bright reported. I think I can explain this to you in terms of kinetic modeling, but let's look at that. Well, at any rate, the results of the Monterey conference were that all of the organ systems were thoroughly evaluated, recommendations made for appropriate ways to measure the effect of dialysis, and 2 primary dialysis therapy kinetic models were considered. The dialysis index, which was essentially believing that uremia was due to larger solute middle molecules poorly removed, and urea kinetic modeling based on urea and small molecular weight solute. The dialysis index had been developed in Washington by Les Babb, the engineer, and Scribner, the physician, and essentially it was time dependent because they were so poorly cleared. A major determinant of dialysis was the treatment time. It's interesting, you see this conflict still rages, and now there's a resurgence of interest in long treatment times and more frequent dialysis. At the same time, Lee Henderson and his engineers had developed dialyzers to do hemodiafiltration and saw some interesting results. And they were campaigning for a study that would look at the effect of middle molecules in dialysis. Ben actually had coined the term high flux membranes, and he made in Walnut Creek membranes that were far more permeable. In the molecular weight range of beta 2 or vitamin B12, which was the arbitrary solute marker we had, they were 5 times as permeable as the membranes in use. So it was like, one of those kidneys was like putting on 5 conventional kidneys and running them simultaneously during dialysis. This is one of the huge advantages of bringing in engineering to this field. You have the opportunity to study such remarkable differences. And this is an example of dialysis with those membranes. They were so water permeable that you couldn't really pick the pressures accurately with the machines we had. So we had to determine the pressure with a burette to measure the rate of ultrafiltration and do that every hour during dialysis. So this was, I'll tell you, very, very tedious work. Nora may remember some of that at Franklin Hospital where we were doing this in the latter part of that time. Well, these are the 2 models. The dialysis index and hemodiafiltration was compressed to time. We were going to look at time as a surrogate for that, and urea modeling. Those are the 2 plans to look at. Now, the way urea was used to control dialysis at the time of the NCDS, it was simply used as a way to try to maintain a constant BUN. So we were thinking of urea as a toxin, even though there was very contradictory evidence to that effect. And so the dose and blood flows and clearances were adjusted. This would be values on 1 month, and this would be means for the group of patients. They would mainly go high and low, and we were doing a lot of dietary counseling to bring these up and these down. But diet counseling guided by urea kinetics, which made it quantitative. So this is how the final study worked out. We had patients distributed into a high BUN group, 110 to 120, and a, quote, low BUN group, 60, 70 to 80. And in both groups, there was short time and long time. So there were essentially 4 arms of this study in terms of both treatment time and diffusive removal of low molecular weight solute. So this, of course, was thought to be with the worst outcome, and the low BUNs would be the best outcome. Now, this was another fascinating slide when we saw it. Here you see the distribution of the patients in terms of whether they failed the study, were judged to become sick or not. And they're plotted here as a function of PCR and BUN. And in the patients with PCR protein catabolic rate greater than 80, they distributed pretty much as we expected. The probability of failure in the high BUN groups was 52%, the probability of failure 13% in the patients with low BUNs. But something was going wrong here. Irrespective of the BUN, the patients with low protein catabolic rates virtually all failed. So there was a 5th group that we had not anticipated, and how does one interpret that? Now, fortuitously, at the same time as all this was going on, we had a patient, Esther Thorson, who I'm sure our nurse would remember, whose BUNs, Nora, were all over the map. Here we were doing urea kinetics, and 1 month they'd be 120, the next month 50, even as low as 40. How could this happen? What was going on to result in such variability? So the only thing I could think of was generation, and plotted the data now, the monthly BUNs as a function of PCR, and you can say it's virtually a perfect fit. And the K2V, the slope of that line, was 1.00, which is what we were prescribing, what we were targeting. So this made a huge impression on me of how to think about this. And so what we did then was simply solve the urea equations over the wide range of PCR and a wide range of BUNs. And the determinant, of course, of the lines was a single pool KT over V. So this was the universe of dialysis dosage according to KT over V in terms of solute and in terms of generation of solute. We then went back and looked at the NCDS data, and all of these patients in this puzzling group, in terms of the dose of dialysis expressed as KT over V, had very low doses. We throttled back the dialyzer and the blood flows if the BUN was low. It was as simple as that. Using the BUN was a totally inappropriate way to guide the therapy, because it led, up in here it's a pretty good way, but down here it led to inappropriately low doses of dialysis and uremia despite low BUNs. So this is the way we ended up interpreting the study, that this was the range of inadequate dialysis. An adequate dialysis with a single pool KT over V greater than 1. And this was in 1985, and this was our modeling line. We could now go back and look at Bostic and Bright in the mid-1800s. Bostic, you remember, we would say that almost certainly, this is PCR, but almost certainly Bostic's patients had far advanced renal failure, were very sick with a BUN of 80, which would have corresponded to a low PCR or a low, particularly a low KT over V. Wright, undoubtedly, I believe, must have had a group of patients that were in early uremia, had no appetite impairment, had good nutrition, good protein intake, and were out here with high PCRs and high, quote, KT over V or residual KR, or residual renal clearance. So I think it could explain the controversy that was 200 years old. Now, this has changed a lot, and as you know, the hemo study was actually taken away by the statisticians. They had convinced the world that the outcome increases ever increasing KT over V, so that they never studied anything below about 1.4. And every year you'd find that there was a higher dose required. The hemo study, where there was a randomized, prospectively controlled study, showed a single pool KT over V in the range of 1.2 to 1.4. Nothing happened above that. So I think we have a dose, we have a rule there, but I think that we don't need to give as much dialysis as we are now, and that critical area was never studied. So I think we've gone as far as we can in terms of urea, treatment time, and dose, and what we need to start thinking and modeling are solutes with real inherent toxicity, such as sodium, calcium, phosphorus, and hydrogen ion. That's it. Thank you. Now, let me say, if there are questions, I will have to have someone tell them to me because I won't be able to hear you.
Do we have any questions? Yes.
Frank, it's great to see you. You were involved with the FHN study, because I saw
You were involved with the
Some of
FHN
your-
study.
The what?
The FHN study you were involved in.
Do.
because I saw some of your comments in the emails that would circulate. What is your take on the value of frequent dialysis?
I think that has real promise. And you can model that and show that you can achieve a great deal more by increasing frequency than by lengthening time. Because as you lengthen time, or as you increase the dose, you're on the flat part of the curve. But if you go more frequent, you get up on the steep part of the curve and you can do a lot more. Now, and as you, I'm sure all know, that the frequent hemodialysis study is now being published and made a big splash at the ASN this year. And there was improved outcome and decrease in left ventricular mass in the daily trial, where they compared short 2-hour dialysis 3 times a week to 2 or 3 times an hour dialysis 6 times a week. But interestingly enough, the initial analysis shows that the one that was thought to be, a slam dunk, that we didn't even need to do the study, was the long nocturnal dialysis, was not better than conventional dialysis 3 times a week. It was not significantly different. And we're in the process of being able to analyze some of that data. And I'm very certain that that's because they allowed too much residual renal function in the nocturnal trial. these were very special kinds of patients. It was very hard to randomize, to get patients to volunteer for 6 to 8 hours of dialysis 6 times a week. And so that if you look at the weight gains and the interdialytic weight gains between the conventional arm and the long nocturnal arm in that part of the study, there was very little difference. And I think it tells me, and we hope to be able to, and we will have access to all of that data to look at the area under the curves of overhydration. And I think it will show that the real factor was the difference in volume control, extracellular volume control. But I do think that more frequent dialysis is useful, and I think you can do a very good job with every other day dialysis, for example, which is much easier, but eliminates that longest interdialytic interval. The closer we can approach some continuous functioning device, the better. And that's why I think the next huge step may be an implantable or a wearable device.
So I have a question. So early in your career, how come you were so sure that this would work for people who are picking research careers? How did you?
I wasn't. I wasn't at all. But I was fascinated with the quantitative aspects of it. I got in, nephrology didn't exist. It wasn't recognized as a specialty when I was an intern and resident. We had salt and water metabolism. And that really turned me on because those are things you could quantify and measure and calculate. And that's what really got me involved in this, because it was very uncertain that it would work. I agree. So I'm a math junkie, you might say.
Was there a change in the materials going from the keel to the hollow fiber dialyzer? And how were those initial prototypes made? And how were the fibers actually synthesized?
Was there a change in the material going?
Yes, it was regenerated cellulose. It was, in fact, that's a very interesting question, because when we had these first dramatic tiny hollow fiber kidneys, their permeability to larger molecular weights was lower than the conventional membrane in the keel dialyzer. And so that's what one of the big arguments that were going on then is that this is very efficient for low molecular weight materials, but the middle molecule clearance is not any better. Now, subsequently, of course, the membranes were opened a great deal, and we were even able to do, as I showed you, very high flux dialysis with them. But the first advent of that technology was with tight membranes.
Can you help us understand the value of the amount of time spent on dialysis versus the actual clearance that you're achieving, and in particular how that will help to determine how much dialysis patients are getting daily dialysis needs?
So the relative value of small molecule clearance versus time, and how that impacts on daily dialysis choice.
Well, time does not do that much, I think, in terms of low molecular weight clearances, but frequency does a lot. I think that there is a tendency now to go back to the gold standard of a 4-hour dialysis. I don't think there's any evidence to support that in kinetics or clinical experience. So I think that what is the time now, Dave? It's probably around 3 hours, 2.5 to 3 hours, or maybe 3.5 hours. But I don't think you can go any further in terms of urea time and schedule other than more frequent dialysis. And I personally think it's time to focus on calcium and phosphorus, which I think is being very poorly handled, and hydrogen ion and sodium, particularly sodium. The idea of sodium modeling has been so bad in loading salt into patients. I think that's, along with a wearable kidney that could give you really continuous clearances, are where the future lay.
Do you not think then that middle molecule clearance is important?
Do you not think that middle molecule clearance is important?
I think we've been trying to prove that for so many years, and we've never really succeeded. There was a paper at the International Congress this year where they showed that a greatly improved outcome with high volume hemodiafiltration, if they stratified the patients by ultrafiltration volume and clearance, obviously. That's such a terrible trap, because that's repeating what DOPS has done for so many years, of looking at the dose predicted, stratified by dose. There's so many traps you get into there. The patients that can't achieve that dose have other confounders that aren't recognized that go with that. That's why the frequent dialysis study, or the hemo study, was so valuable, showing that ever-increasing outcome doesn't occur. So I suspect that there are middle molecules. I expect that there are important ones, but I don't think we have a real good way to study those at this point. And so far, they've proven to be very elusive in terms of playing a major role in toxicity of uremia.
What do you think is the effect of shorter and longer dialysis as far as volume removal is concerned? The pathology of trying to remove a lot of fluid in 3 hours.
So the issue of short dialysis time and sodium and volume control.
Oh, there is a very, very important interrelationship, I agree. And when I think of shortening time, I think of achieving optimal fluid balance without symptoms, so that you may well have patients that simply don't tolerate high rates of ultrafiltration, and the time is going to be determined by volume requirements, without doubt. That's something you can compromise on. And so I would say that's very important. And that's why I think the nocturnal arm of the daily dialysis study failed, because they had so much residual renal function that the conventional arm didn't have significant fluid overloads.
Can you model volume removal versus pathology?
Yeah, you should be able to. And we hope to do that as one of the things, if I live long enough, that we're interested in. The problem there is that we have very poor control over dialysate sodium. And the problem is not the machines. The machines, they're all volumetrically proportioned, and the tolerances are tight. But manufacture of dialysate it varies as much as 2.5%. That's just too much, and that has to be improved. Now, we think that it's possible, if we ever get all the methods together, that you could teach the machine how to compensate for errors in concentrate by looking at a family of conductivity curves and adjusting. I don't want to go into it in detail, it's too complicated. But I think you can teach the machine to do a much better job of prescribing the sodium you want. If we can do that and get it to within a milliequivalent per liter, then I think some models could be developed. We have developed models. For example, what I think you should do, if you're going to do any kind of modeling, is at the beginning of dialysis, when the plasma volume or body water are high, that's when you should use a low sodium, because you can take out convectively and diffusive salt at the time they're overloaded, and then the dialysate sodium should come up as you go down, and maybe even go above if you've had convective loss. But that's not the way it's done. It's been done just the opposite way. But I think there is a future for that.
I'm going to finish with one last question.
Yeah.
So, any advice to young fellows starting their career about what they should pursue in terms of...
I'm not a person that asks that question too. I've been at odds with the establishment all my life. I think that you should do what's most interesting to you. I think that's always going to be the case. But if you want to go into this field, be sure you hook up with a good engineer.
Thank you very much.